what does it mean if you have a trait that when you take two parents that differ in the trait, their kids have all sorts of variations in that trait? What are the two options that we have? It's either, there are two explanations for why you see such variation. What do you think? Yeah?
Sorry,
That's okay.
Incomplete dominance?
So some sort of dominance effect, so one could be incomplete dominance. What's a good example of incomplete dominance? We didn't talk about this in class. There was an example in one of the chapters, though.
Pink rose.
Right, there's some flowers where if you cross a red flower and a white flower, you get a pink flower. Incomplete dominance. Okay. What else can affect phenotypes besides dominance? Obviously, today we're talking about complex traits. That's another answer. So if you have lots and lots and lots of genes that all together have to sum together to produce the effect, that's another reason that kids don't necessarily look like one parent or the other or halfway in between. What else affects our phenotypes besides genetics? I know this is a genetics class.
I was going to say, like the linkage, but you say that sometimes that some children come out looking a lot like the parents.
Right, so how much you look like your parents can depend in part on linkage. So are there a bunch of traits like earlobe attachment and nose shape and so forth that are all inherited together as a package, so you look more like one parent than the other? The other answer, environment. Is it nurture or is it nature that affects our phenotypes? Skin color, certainly. If I go out to the beach, which I've done in the past year, although you can't tell, and I don't put on sunscreen, my skin's going to change color, but that doesn't have anything to do with my genetics. That just has to do with an environmental effect. I got a sunburn. And for all of these reasons, we have to be really careful. We're not going to get into too much detail, but you have to be careful to distinguish what's an environmental effect from what's a genetic effect. When you look at parents, their offspring, and you see differences in the offspring, how do you know it's genes? How do you know it's not just lifestyle? And that brings us to one of the first points we'll talk about, which is about twins and twin studies. Is anybody in here a twin? You don't have to share if you don't want to. You've got one in fact. I know some twins. I'm not a twin. But twins are really useful for understanding the differences between genetic effects and environmental effects, and we're going to see that in just a minute. So a quick review from last class. We were talking about the breast cancer gene. One of you asked a really important question, which was, if the breast cancer gene, which is involved normally in DNA repair, is in all of our cells to save us from DNA mutation, then why is it only predisposed to breast cancer? And I didn't know the answer. So where do you think I went? Oh, don't look at the bottom line. Where do you think I went to look for answers? I went to OMIM, Online Mendelian Inheritance in Man, and I typed in breast cancer. Let's see what. So one of the answers here is, A, scientists really don't know. So this is my Cliff's Notes from reading the OMIM entry. Why BRCA1 mutation predisposes mainly to breast and ovarian cancer isn't clear yet. It has been shown, and this is just one example, the second bullet point, that males that have mutations in BRCA1 might be predisposed to prostate cancer. So it might not just be a breast tissue specific effect. It might just be that we don't know enough about how this gene affects other tissues yet. And to prove my point about polygenic traits and complex traits, let me show you what OMIM.org says about breast cancer. So you could do this too if you like. Just go to OMIM, type in breast cancer, see what comes up. There's a long list of entries in Online Mendelian Inheritance in Man on breast cancer. That's because there are lots of genes involved in producing breast cancer. If we select this top link up here, the one with the pound sign next to it that just says breast cancer, this first table says phenotype gene relationships. It's a heck of a lot of genes. So we've got RAD54L on chromosome 1, CASP8 on chromosome 2, BARD1, chromosome 2. This is a huge list of genes, all of which have been reported in literature to be involved in producing breast cancer. And it goes on and on. Scroll down, scroll down, scroll down. There's like 40 or 50 genes that have been reported to be involved in whether or not you're susceptible to breast cancer. It's a great example of a complex trait. Just because you have one mutation, say in BRCA1, does not mean you will get breast cancer. The more mutations in more of these genes you have, the more likely you are, perhaps, to get breast cancer. It's the essence of a complex trait. Breast cancer depends on a lot of genes producing that phenotype. We'll talk a little bit more about complex traits today. Any questions at this point? Complex traits are traits that involve multiple genes that interact together to produce a phenotype. Hopefully everybody read for today's class this really cool story. It wasn't very long. It scrolled forever and forever and forever because there were lots of cool pictures of twins that don't look like they're the same, non-identical twins. What types of twins do we have? Right, so we've got fraternal twins, which we also call, so we've got non-identical twins, which in this class we're going to call dizygotic twins. And then we've got identical twins, which are monozygotic. What sort of twins are we looking at in this picture? Are these monozygotic twins or are they dizygotic twins, do you think? I think if you read, which I'm sure you didn't, if you read the comments that were way down at the bottom of the article, somebody pointed this out. Are these identical twins or non-identical twins? Non, clearly. They're not identical. So here we're talking about looking at cases of dizygotic twins. So what's the difference between the two types of twins? Hopefully this is review, but it doesn't need to be. How does a dizygotic twin form? Or how do dizygotic twins form? Right. 2 sperm, 2 eggs. Hence, how identical are dizygotic twins? They're right, they're brothers or they're sisters, or, as in the first pair of twins I knew, they're brother and sister. So this is when mom happens to ovulate 2 oocytes at the same time during the same menstrual cycle, and 2 sperm happen to independently fertilize those 2 eggs. So you get non-identical twins. Of course, they're as identical as any brother and sister. So they probably share a lot of the same traits. They might have the same body shape, eye color, hair color, whatever. But they're not as identical as the monozygotic twins. So how do the monozygotic twins form? How many fertilization events? Mono. 1. So what happens after fertilization? Right. At some point during the early cell division process, the embryo splits in half, not necessarily exactly in half, but it divides, and each of those 2 small groups of cells independently forms an organism. So you get 1 fertilization event that leads to 2 organisms. So you can imagine something like this. You get each of those gametes carries 1 copy of each chromosome, so you get a diploid individual. Divides into 2, divides into 4, and then that mass divides into 2 separate groups of cells, each of which goes on and forms a twin. through additional cell divisions and development. And they're identical because they were all fertilized, one sperm, one egg. All of the DNA from there on is identical by DNA replication, which we've all seen.
So, why does the oocyte split up?
It's not, okay, so it's not the oocyte, good question. It's not the oocyte, but rather at some point during cell division, right, at this 4-cell stage, for example, somehow this group of cells falls apart. So I'm not very clear on the mechanics of how this works. We just know empirically that it happens because we observe identical twins. So we know at some point during early development, when there's a really small number of cells in the fetus, it divides somehow. The groups of cells split into 2 groups or more, identical triplets, identical quadruplets. But the forces that cause that aren't well understood. Yeah?
Is there a mechanism in place that tells like the now 2 separate groups of cells that they need to undergo more divisions because now they're making 2 of everything?
That's a really good question. So does the developing embryo have a way to sense that, wait a second, I don't have enough cells. I'm so far into the developmental process already and I've got half as many cells as I'm supposed to. Do I need to divide more or not? What would happen if the embryo didn't have such a mechanism?
You wouldn't have 2 viable fetuses.
You might not have 2 viable fetuses. What would we expect twins to be if they didn't have that sort of a mechanism? If they were born? What would twins look like if the embryos didn't have a way to detect how many cells there were? They'd be smaller, right? You might expect twins to be half the size of a normal adult if the embryos didn't have a way to figure out, how many cells do I have? I need to divide more. So there is a mechanism, but I'm not sure. Yeah, that's a good Biology 155 for those of you that are interested. Developmental biology. We go into a lot more twin-related and development in general questions. Excellent questions. Others? So this is really cool. So twins are different. Look, here's a pair of identical twins. One of them is up in space right now. In fact, if some of you are following on Twitter, this morning I learned that he's the commander of the International Space Station, has now broken the US record for a number of days in space by a US astronaut, right? Mark Kelly. So are they identical? Look carefully. Look at the caption of the picture. Are they identical? Yeah, except for, what does the guy on the left have that the guy on the right doesn't have? A mustache. So they're not identical. Wait a second, but they're monozygotic twins, so how are they not identical? Is it nurture or is it nature? It's environment. Well, unless one of them actually doesn't have the ability for facial hair. One of them just chose not to shave, and so they look different, right? It's an environmental effect. It's behavior. So there you have behavioral differences. They're not completely identical twins. So right now, this is a fabulous way for scientists to get information about how our genetics affects our behavior and affects our traits in general. We have one identical twin who's been in space for over a year. Weightlessness, solar radiation, and so forth. And we've got his twin brother who's been on Earth the whole time. So what did the scientists do?
Maybe one of them could age less when he's out in space.
Yeah, that's true. So they took samples of both of the twins before Mark got launched to the space station. And they've been taking his samples and his brother's samples the whole time they've been apart in different environments. And then at some point, Mark comes back to Earth, and they take more samples and do more tests, and they find out, is there something, do gene expression changes, that is, do the types of genes that are turned on when you're floating in space for a year, as opposed to being on Earth, change? So can we understand anything about how our environment affects our genetics, affects our physiology? So they're going to compare the two genomes and the types of genes that are turned on and genes that aren't turned on and so forth. But like we talked about in class before, what's the issue here? They're going to have their whole DNA, their whole genome sequenced. Right, so what if they happen to learn something that they don't want the public to know? So this is a, what does it say, $1.5 million study. It's been funded by the US government to do this sort of test. It's a great experiment. There's an experiment up in space, there's a control individual down here on Earth. But the results might never be published, as this says. So what if they learn that they both have predisposition to something they don't want their insurance company to know about? If you're an astronaut, maybe you don't have to worry about insurance companies. You'd hope that maybe the US government would give you some, oh, maybe not. It's not a political science class anyway. Who knows? Right. So this is one way that studying twins, now we have to do it ethically. Informed consent, right? This is not the 1800s. You can't go like steal some twins and hide them away in the forest and do weird studies on them. You have to get informed consent. You have to tell the twins, of course, exactly what you're planning on doing, what the risks are to them, what the possible benefits are, and let them decide do they want to participate or not. But it's a great way to get information about how genetics, because they're identical, it's a great way to know how environment affects our traits and how genes can affect our traits. So we've already started filling in this little summary of this part of the class today. So how do dizygotic twins have different phenotypes? What are the ways that that can happen?
Different genders.
So they've got different, and what is the basis of their different genders? So dizygotic twins can be male and female separately.
They're getting different genes.
Right, so they inherit different DNA. They inherit different genes because it's two different fertilization events. So there can be a genetic basis to non-identical twins being non-identical. They're just as identical as any brother and sister, or brother and brother, or sister and sister would be. What else? We've got genes and we've got environment. What about the identical twins? What can make them different besides environment? So environment is always possibly causing one twin to have a mustache and one not. Is there any way genetically that identical twins could be different?
Mutations.
Yeah. Mutations. So once that initial group of cells divides into two organisms that develop independently, if a mutation that happens randomly happens in one and not the other, sure, there are still genetic differences between identical twins. Probably not very many genetic differences between identical twins, but they will exist. I don't suggest you go tell your identical twin friends this. They might not appreciate it. Did you know you're not really identical? They might enjoy it. Who knows? Okay. So now we can get into a little bit of quantitative traits, unless anybody has any questions about twin anything. Again, the bottom line, the basic point is that we all differ because of both genetic and environmental effects. Twin studies can help us distinguish which effects are genetic and which might be due to our environments. Okay. So quantitative traits then. Aside from whether or not you can grow facial hair, one of you mentioned skin color this morning, which is a great example of a quantitative trait, as in height, eye color, hair color, etc. So I want to start off with this relatively simple example. how quantitative traits work and how you can identify a quantitative trait. So let's assume that we're working with an organism that has at least 3 genes that controls skin color together. So every time you have a capital allele, as it says up there, every time you have a capital allele at one of these 3 genes, you get a slightly darker skin color. So if we compare the 2 parents, how many capital alleles does the parent on the left have? 4. And on the right, 2. So which parent has darker skin? Yeah. Parent on the skin has more dominant alleles that increase the amount of pigment in the skin. So parent on the right has 4 capital letters, darker skin. The parent on the right has 2 capital letters in their genotype of these 3 genes, lighter skin. So these are the questions that are really important to discuss for an introduction to quantitative traits. When these 2 individuals that have 3 genes that control this trait have kids, how many different possible skin pigmentation levels or shades could be produced? So I'd like you to be able to predict in this cross, for example, which we're going to do right now, what sort of traits were you expecting to see in the next generation? Do all the kids look just like the parents, or there's going to be a variety? So if we're going to do this, what do we have to set up? Right now. The P word. You know, like by the end of the term, you'll be saying it's a, you know, oh, you said the P word. How do we develop a Punnett square? We've never done this before. 3 loci. There are the genotypes. We've got gene A, gene B, and gene C. Is it a square or is it a cube? Punnett square or Punnett cube? How many parents are there in this cross? 2. So there's going to be 2 axes. Right. One's got the genotype, the haplotypes of one parent's gametes. The other axis has the other. So what are the different haplotypes of gametes that these parents produce? Let's do the parent on the left first. You've got 3 genes, you've got 2 alleles of each. Well, it's not quite true. What's every gamete going to have at the A locus? So there's going to be a bunch of, everybody's going to have a capital A. Okay. What fraction of the gametes are going to have a capital B? Half. We'll have a capital B, half will have a little b, and then what about the C? Same thing. So every time there's a big B, there's the possibility of having a big C or a little c. Every time there's a little b, there's a possibility of having a big C or a little c. How can I make this more challenging? What would I have to change in that first parent's genotype if I wanted you to do even more work? Right, change the first gene to being heterozygous, big A, little a. So if it was big A, little a, big B, little b, big C, little c, you'd have to do 8 different haplotypes down the left side. Okay. So we've got 4 different haplotypes produced by the parent on the left. The same is true of the parent on the right. Everybody gets a little c because this parent happens to be homozygous for little c. So then it's big A's with big B's, big A's with little b's, little A's with big B's, little A's with little b's. Yep.
Is there like a 2 to the end formula that tells you how many haplotypes you're going to get, or you just have to do all the different haplotypes and then see how many you get?
Oh, you want a formula? Does anybody know? It's a great question. It's really useful if you want to know exactly how much time you have to allot to doing a problem during an exam. So if you have 2 alleles at every gene, and 2 alleles at the second gene and 2 alleles at the third gene, then it's essentially 2 to the number of genes that you're talking about. This is for a diploid. If you were tetraploid, instead of a 2, it would be 4, and so on. So how does this work? Let's do this for one of these examples. Let's do this for the parent on the left. How many different possible haplotypes could be produced? Gene A, how many alleles are there? How many different versions of this gene are in this individual? Just the one. Then we've got 2 alleles. We've got 2 alleles there. So you just multiply them all together. You get 4 possible combinations. If every one of 3 loci is heterozygous, then you've got 2 times 2 times 2, or 8 possible combinations, 2 to the third. Questions? Okay. The real question is, how many different pigment types are produced in these kids? So instead of filling in the Punnett square, which would be a ridiculous waste of time at this point, let's do something simpler. Where in here is the lightest colored offspring? Which one? The top right or bottom right corner? Bottom right corner. How many capital letters does this individual have? Okay. Where's the most pigmented individual in this Punnett square? Top left, we've got 5. Okay. We've got 4 capital letters in that cell, 3, nope, sorry, it's 4. Sometimes, I hate using C as a G, I forgot, I use C. It's hard for me to tell my big C's and little c's apart. 5, 4, 4, 3 capital letters there. Okay. You fill out the rest, I'll fill it out too. And we'll see what we get when we're done. So how many capital letters are in each square? That helps us predict the pigment type, how much pigment is in each one of these individuals. We got one individual that's got 5 capital letters, one that's only got 1, the rest are somewhere in between. Yeah.
On the top row, the second one from the left, should that be 4 or 3?
So this one, yeah, that's supposed to be a capital B.
And then
So-
we have 3 capital.
So that should be a 4. Thank you very much. We have another winner on Prove the Professor Wrong. Thank you. Yeah, so hey, look, it's symmetrical. Almost. That means that, uh-oh. Hey, look, is it symmetrical? Oh, good. This is what happens when I can't read my own handwriting. What's interesting about these data? How many capital letters did the first parent have? 4. and this other parent had 2. What do their kids look like? Do the kids look like the parents? In terms of skin color, how dark their skin is. We've got some kids that have 4 capital letters. We have some kids that have 2 capital letters, as we've just filled in this table. What shows up this time that we've never seen before?
5.
A 5 upper left and a 1 in the bottom right. This is called transgressive segregation. What transgressive segregation is simply put, when your kids have phenotypes that are more extreme than either of the parents. They transgress or they cross the boundaries of the phenotypes that the parents had. So in this case, parents with somewhat dark and somewhat light skin color have a possibility of having a huge range of skin colors in their kids, including kids that have darker skin than they do and kids that have lighter skin than they do. And transgressive segregation is something that you usually see when you're dealing with a quantitative trait. A trait that has more than one gene that contributes to the overall effect, the overall phenotype. You have the possibility that you have intermediate parents and that their kids have more extreme phenotypes than the parents do. It'd be like Mendel crossing a green pea and a yellow pea and getting a purple pea or something like that. Like where'd this come from? Shouldn't it have been a yellow-green or a green-yellow pea hybrid? Right? When new types of phenotypes, that's often transgressive segregation. So another way to look at these data would be to plot a histogram, and that's the last thing I want to work with you with today, is making a histogram out of the data we had in that Punnett square on the previous page. So on a histogram, we need to have, what goes on the y-axis on a histogram? We're looking at histogram. Frequency, number of observations usually. So how many times you see an individual with a certain skin color, for example. So the phenotypes go on the x-axis. So we'll just say individuals with 1, 2, 3, 4, 5, 6 capital alleles. So skin color goes from lightest on the left to darkest on the right. How many observations did we have for total capital alleles of 1? I think it was just that one kid in the bottom right corner. So we see one individual that has 1 capital allele. 2, 3, 4, 5, 6. Right, so we're just categorizing how many times do we make these observations of different phenotypes. What about how many individuals had 2 capital letters on that table? Four. Okay. What about 3 capitals? I counted 6. And if that table's symmetrical, what's going to happen to data point for 4 capital alleles? It's going to go back down just because it was symmetrical. There's one individual with 5, no individuals with all 6 capital letters. So you can connect the dots and you get something that looks sort of like a normal distribution. There's continuous variation in this trait. So quantitative traits are sometimes also called continuous traits because there are not distinct or discrete versions of this trait. There's not just all really dark skin and all really light skin. There's every shade in between, essentially. And again, these sorts of histograms, when you see them, when you see a continuously varying trait that's got some mean average in the middle that's most frequent, that also suggests when you're dealing with a quantitative trait, or a trait that's controlled by lots and lots of genes. So a quantitative trait could be 2 genes, it could be 3 genes, it could be 4 genes. Like breast cancer, it could be 20 or 30 or 40 different genes. Like human height, it could be hundreds of genes that affect that trait. Any questions? So we predicted the offspring of a cross involving a quantitative trait, lots of loci, 3 loci at once, Punnett square, predicting the phenotypes of the offspring, and then building a histogram to look at the frequencies of those traits. So in sum, don't bother with the first question. How many different genotypes? That involves actually filling in the Punnett square and counting genotypes. I know you could do that. Don't bother. But how many different levels, how many different iterations, how many different shades of skin color were produced in this cross? We had 1s, 2s, 3s, 4s, and 5s. There were 5 different categories, 5 different groups of how many capital letters did you have in your genotype. And then what was the term that I used for this last point? That when the kids' phenotypes extend beyond the range that the parents had, that's transgressive segregation. So that's the last bullet point. And that's always a clue that we're dealing with a quantitative trait. So to help us prepare for the exam on Friday, I've left 10 minutes in class, although you don't have to spend the time now doing this necessarily. I would appreciate you thinking for 10 minutes about what sort of questions I'm going to ask on this test, like I always do. So I'm going to send out, just a second, so I'm going to send out a Google assignment that asks you to voluntarily participate in this. Spend 10 minutes now if you've got the time. You do have the time. Spend 10 minutes right now, voluntary, no points attached. Think about a question that you think I will ask. These are the topics that are going to be on the exam. We're going to include the chi-square test this time. Genetic mapping, dominance, and quantitative traits are the chapters we've covered. Sir?
On the last slide.
Yes.
So if you would not get a pigmentation that's higher, let's say pigmentation values would have been from 4 to 1, would that not be considered transgressive?
Right.
Because it's between the values of the parents.
Right. So if the kids, even if the kids have the same phenotype as the parents, that does not count as transgressive.
You need something over.
It has to be beyond the range of what the parents' phenotype, whatever it was, looked like. Eye color, skin color, height. And we see this a lot in crosses in nature, which is kind of cool. But it also means that genetics is complicated because most traits are quantitative traits. It makes it a lot harder to study how they come about.
You said for it to be transgressive, it has to be over what the parents are, but if it's under?
Over or under.
Oh,
So
over or
beyond
under.
the range. So if a trait is less than than both parents or greater than both parents, that counts as transgressive. Yeah. Okay. So next class we're going to do study guide distribution and review session, so come to class with questions, please. I'm going to try to, if you help me get these questions to me today, I'm going to try to get the study guide out early in the morning tomorrow, so that you've got a day at least to look over it and come back with questions. Other than that, please work on this, but that's it for today.